Good afternoon. I'm Parveen Yaqub, Deputy Vice-Chancellor and Pro-Vice-Chancellor Research and Innovation at the University of Reading. On behalf of the Biochemical Society and Portland Press, I'd like to extend a warm welcome to today's webinar, which is the first of the year and part of the Biochemistry Focus webinar series. So this series will include a range of topics in molecular biosciences, as well as some practical sessions aimed at supporting career development. There'll be lots of opportunity to ask questions, and we'd really welcome any suggestions that you have for future topics and speakers. So take a look at the website for details about how to do that. Today's webinar is on the theme of the importance of essential fatty acids and their derivatives, and it was inspired by a themed issue on lipid mediators, which was published in 2020 in the Portland Press journal Essays in Biochemistry, which was guest edited by John Harwood, one of today's speakers, and his colleague Emir Lloyd-Evans. Lipid mediators are biologically active metabolites formed by enzymic and non-enzymic oxidation of polyunsaturated fatty acids. And the issue explored the many physiological processes that they're involved in, as well as how they become dysregulated during aging and in lipid-related diseases such as diabetes, atherosclerosis, and metabolic syndrome. The special issue was dedicated to Professor Mike Wakelam, who was one of the giants of the lipid signalling field and who sadly died in March 2020. So the editorial in that issue describes his work and influence, and it's worth taking a look at the journal because it offers reviews from experts on key topics in biochemistry, written to be really accessible to those who are not immersed in the subject. So do take a look at recent issues, especially the molecular mechanisms of neurodegeneration, which was published just before Christmas. And you can visit the journal's website for more details at portlandpress.com/essaysbiochem. So during today's session, we're going to hear from Professor John Harwood from Cardiff University and Professor Philip Calder from the University of Southampton, who will be discussing the sources of essential fatty acids, the lipid mediators derived from them, and their functions in health and disease. Each of the speakers will give a short presentation and then we'll take questions for both talks at the end, but please do send your questions in during the talks, making it clear who the question is for. And you can do that by typing in the question box, and I'll be sifting through the questions as they come in, and I'll try to put them, as many of them as I can, to the speakers at the end. So don't be shy, there's no such thing as a silly question. So it gives me great pleasure to introduce our first invited speaker today, Professor John Harwood from Cardiff University. John works on the metabolism and function of acyl lipids, which has involved the discovery of new pathways, characterization of important enzymes, and the use of metabolic control analysis to quantify regulation. His research touches on environmental aspects as well as medical considerations, and he's published research on many different organisms, but mainly on algae, higher plants, and mammals. He has expertise on the biosynthesis of polyunsaturated fatty acids in photosynthetic organisms and the health benefits, especially in brain function. So over to you, John. And we can see your slides fine, John.
Okay, so between the two world wars, it was thought that lipids in the diet were only used as a calorie intake and nothing much else. And pioneering work by George and Mildred Burr in 1929, where they fed rats a diet which didn't contain any lipids whatsoever, revealed that, in fact, lipids were important for good health and not just as a source of calories. And further work by the Burrs the following year identified two essential fatty acids which couldn't be made by mammals, but were essential for good health. And the basic essential fatty acids are linolenic acid, or alpha linolenic acid to be precise, which is an 18-carbon fatty acid with 3 double bonds. And the first double bond is 3 carbons from the methyl end of the hydrocarbon chain. So this is referred to as one of the N3 or omega-3 fatty acids. The other essential fatty acid was linoleic acid, 18 carbons with 2 double bonds, and the first double bond is 6 carbons from the methyl end of the hydrocarbon chain. So this is an example of the N6 or omega-6 series of essential fatty acids. Now, although these are both essential fatty acids and are essential for our good health, they don't themselves actually do very much in terms of health benefits. This is a generalization. There are specific beneficial effects of these two acids on skin health, for example. Next slide, please. The reason that essential fatty acids have all their very important effects is that they are lengthened, chain lengthened to 20 and 22 carbon polyunsaturated fatty acids, such as arachidonic acid, which is a 20 carbon fatty acid of the omega-6 series. And these fatty acids are released from membrane lipids on stimulation, and they're then oxidized by a series of enzymes, the lipoxygenases or LOX enzymes, the cyclooxygenase enzymes or COX enzymes, and the cytochrome P450 epoxygenases. So these oxidizing enzymes then metabolize the various polyunsaturated fatty acids to produce a whole series of lipid mediators. And it's these lipid mediators that actually have the particular action on the body. And for example, prostaglandins were the first ones really that were identified. Prostaglandin E2 is produced from arachidonic acid, and it gives you a headache if it's produced in the brain. So if you've, dare I say it, have drunk too much and got a hangover, you'll be producing prostaglandin E2 in your brain, and you'll have a terrible hangover and terrible headache. So what do? You take an aspirin or ibuprofen to relieve the pain, and what these compounds do is they inhibit the cyclooxygenase, which is acting on arachidonic acid to produce prostaglandin E2. So these lipid mediators then produce acute effects at very, very low concentrations in the tissues that they're actually produced in, i.e. in the brain if you have a hangover. And they're produced at levels of 10 to the minus 8 or 10 to the minus 9 mole. So they're producing their effects at very low concentrations indeed. Now, it's very seldom that people actually suffer from essential fatty acid deficiency because you don't need very many essential fatty acids in order to alleviate or prevent you getting the deficiency syndrome. However, it's been recognized over the last 20 or 30 years that these lipid mediators produced from polyunsaturated fatty acids have all sorts of very, very important effects. And furthermore, it's because, as I've said before, it's the 20 and the 22 carbon fatty acids that actually are converted to these lipid signaling molecules, then the level or the generation of these longer chain polyunsaturated fatty acids inside our bodies is very important. And that could be taking them in the diet. And so there's been increasing interest recently in taking in sufficient of the very long chain polyunsaturated fatty acids in our diet in order to have good health. Next slide, please. And this shows a proposal from Steve Cunane, which was made a number of years ago, talking about all the different members of the omega-3 and the omega-6 series, the various fatty acids, and some can be considered essential under certain circumstances, but not under others. So in adulthood, there's only one conditionally indispensable polyunsaturated fatty acid, and that is alpha-linolenic acid. Whereas under other conditions, such as in pregnancy, lactation, or childhood, other fatty acids become indispensable. For example, the longer chain fatty acids, such as arachidonic acid or docosahexaenoic acid, 22 carbons with 6 double bonds, of the omega-6, omega-3 series, then these become indispensable. But other members of the polyunsaturated fatty acid series can be very useful in the diet. Because there are two series of PUFAs or polyunsaturated fatty acids which are essential, the omega-3 and the omega-6 series, the omega-6 series are normally present in quite large amounts in our diet, so we don't really have to bother about how much or getting extra omega-6 polyunsaturated fatty acids, but we do have to maintain good levels of omega-3 polyunsaturated fatty acids. Now, where do we get these from? Next slide, please. We get these from fish, and various governments in Europe have recommended that we should always have fish in our diet, and furthermore, normally they recommend that oily fish is in our diet. And the UK Advisory Committee, for example, suggests that we should have fish twice a week. One meal would be an oily fish. What do I mean by oily fish? I mean something like herrings or mackerel or sardines, salmon, trout, these sort of fish that contain quite a lot of fat in their meat. And this will provide roughly 0.5 gram of long-chain omega-3 fatty acids per day. Next slide, please. However, one of the reasons for taking in long-chain polyunsaturated fatty acids rather than just relying on the 18-carbon fatty acids is that we are pretty poor at converting 18-carbon fatty acids into 20 and 22 carbon polyunsaturated fatty acids. For example, females can convert, if they take in linolenic acid, they only convert between 3 and 5% of that into DHA. That's the best they can do. Males are even worse. They can only convert 1 or 2%. So it makes good sense to have the longer chain, the 20 and 22 carbon polyunsaturated fatty acids in our diet. And this is where algae come in. Fish can't do it any better than us. They are reliant on eating algae in order to get their longer chain polyunsaturated fatty acids. So algae are important. They're at the bottom of the food chain as a primary source of fixing CO2 from the atmosphere by photosynthesis into various compounds. And they produce polyunsaturated fatty acids in good amounts, including 20 and 22 carbon polyunsaturated fatty acids. They're the source of nutritional fish oils because fish are reliant on the algae that they eat in order to have nutritional fish oils. They're a source of useful enzymes that I'll discuss in a moment. And they're also algae are can be used as feeds for humans, animals, and particularly in aquaculture. Next slide, please. So how do organisms such as algae, and to a limited extent ourselves, convert something like linolenic acid into a 22 carbon polyunsaturated fatty acids such as DHA? Well, we have to use a series of desaturators and elongators in order to do this conversion. And in mammals, the conversion goes by a roundabout route from the 22 carbon stage around here to make 24 carbon polyunsaturated fatty acids, and then by oxidation, beta oxidation, back to DHA. This is the so-called Sprecher pathway, because we don't appear to have a very active, if at all, a delta-4 desaturators that can do this directly. One thing to recognize in this slide is that both the omega-3 and the omega-6 fatty acids are modified by the same enzymes. So there's competition inside us between omega-3 and omega-6 fatty acids. So if you take in a lot of omega-6 fatty acids in the diet, for example, they will outcompete the omega-3 fatty acids and vice versa. So you have to bear in mind the ratio of omega-6 and omega-3 fatty acids which you take in your diet. Next slide, please. Okay, so here's a fatty acid composition of a number of algae. There are estimated to be somewhere between 50,000 and 1 million types of algae. So huge numbers, and it's not surprising that they vary a lot in both their appearance and in the fatty acids that they make. Here are some freshwater species. These are green algae. They produce lots of polyunsaturated fatty acids of 16 or 18 carbons in chain length. This is an abbreviation for individual fatty acids. The first number indicates the number of carbon atoms, and the second number, the number of double bonds. So freshwater algae will produce lots of 16 and 18 carbon fatty acids, but not 20 or 22 carbon polyunsaturated fatty acids. A few salt-tolerant species will produce limited amounts of 20 and 22 carbon fatty acids, but only rather limited amounts. Next slide, please. However, once you go into the marine environment, into seas and the oceans, then you have lots of different species here, and you will see that most of them will produce quite appreciable amounts of 20 carbon fatty acids or 22 carbon polyunsaturated fatty acids. So these, of course, are the source of the long chain polyunsaturated fatty acids that fish consume and that we in turn consume when we eat fish. Next slide, please. So I've already shown this somewhat bewildering pathway which allows you to convert linolenic or linoleic acids into the longer chain polyunsaturated products. And the algae are able to do this direct conversion of 22 carbon fatty acids into DHA, for example, or into the equivalent omega-6 polyunsaturated fatty acid, because they have a very active delta-4 desaturase that removes hydrogens to create a double bond in the delta-4 position. Algae also have an alternative pathway here, which just has the desaturases and the elongasers working at different stages in the overall pathway. Next slide, please. So if you're interested in algae producing polyunsaturated fatty acids, you need to know the sort of factors that will influence this process. And one of the important factors is nutritional status. If the algae are growing and they lack key nutrients, in particular, they lack nitrogen or to an extent phosphorus, and if they're diatoms, they may lack silica, this gives them nutrient stress. And under those conditions, they stop growing, stop making membrane fatty acids, and accumulate triacylglycerol in large amounts. Then when you refeed them with the deprived nutrient, then they will convert the triacylglycerol stores back into membrane lipids and start growing again. So that's one way you can get them to accumulate a large amount of lipid. Temperature has an effect. This is bad news with climate change. Increased temperatures reduce the amount of desaturation in general. So that's a problem or potential problem that's becoming apparent. And then there are various other factors which will affect the amount of lipids that algae are making. Next slide, please. Okay, so a lot of algae, of course, go into the fish industry. And interestingly, over the last few years, a majority of the fish that are consumed in the world actually come from aquaculture. The figure's about 54–55% at the moment. So rather than wild catch, we actually farm fish by aquaculture. And these farmed fish require fish meal, and it sounds ironic, fish meal and fish oil in order to be grown successfully. And a large amount of the fish meal which is produced, up to 2/3 of the total fish meal and about 3/4 of the fish oil that's produced by the fish industry, is then refed back to aquaculture. This is obviously unsustainable, and we have to consider what we can do about this. Next slide, please. And the fish themselves will vary very much in how much oil they have. Oily fish, such as mackerel, sardine, trout, salmon, and so on, have a large amount of oil in their flesh, whereas a lot of the principal farmed fish, such as carp and tilapia, these have very little. And of course, the white fish that we're more familiar with have rather little oil. So, of course, it's not necessary to try and get fish to accumulate a large amount of oil if they're not going to do it, if they're white fish, if you like. Next slide, please. And because of that, we can reduce the amount of fish meal or fish oil that's used in aquaculture. So we can modify the intake with the species of fish that we're actually farming. We can use alternatives to fish, including plants and algae, to provide the oil. And we can use what they call the sparing and finishing diet. So you feed a fish with a low amount of oil or a low amount of fish meal, for that matter, for most of their life, and then it's only before they're harvested that you provide them with a finishing diet, which provides the extra oil in order to increase the amount of oil in the flesh of the fish. Next slide, please. Now, I mentioned in growing algae how you could boost the amount of lipid which was accumulated, and this shows one example where this is Chlamydomonas, a green alga. It's grown with low amounts of nitrogen. In fact, it's lacking nitrogen in its growth and in the growth medium, and it accumulates a triacylglycerol. You then refeed nitrogen so that it can start to grow and to multiply, and immediately the triacylglycerol goes down, and at the same time the amount of membrane lipids, polar lipids, goes up, and the opposite, of course, during nitrogen starvation. So this is a way you can get algae to make more lipid or accumulate more lipid, which you can then harvest. Next slide, please. Okay, so I've described algae carrying out fatty acid biosynthesis, producing palmitic acid, saturated 16-carbon acid, and then using various elongasers and desaturators to accumulate polyunsaturated fatty acids, such as DHA 22,6. There's an alternative pathway here, which uses an enzyme, a huge enzyme, very high molecular mass, which is called PUFA synthase, polyunsaturated fatty acid synthase. It's like a polyketide synthase, which is present for making secondary products in plants. And this will add 2 carbon units all the way through and then produce really just a single product with no accumulation of intermediates. And the product could be, for example, DPA or DHA, with very little intermediates accumulated. So this is a very important and rather useful alternative system. And it's present in organisms called thrustoschistrids. It's a bit of a mouthful, I'm afraid, or I find it a mouthful. This is a group of organisms which were originally designated as algae. They're not algae, they don't photosynthesize, but they're rather like yeasts and fungi. But they're not fungi either. They're a separate group of organisms. So they have an alternative way of making polyunsaturated fatty acids. And one of these PUFA synthases has been put into oilseed rape to persuade oilseed rape to make DHA, which it does. Just small amounts, but it's a proof of concept anyway. Next slide, please. However, the usual way, or the way that's been successfully done over the last few years, is to put all this mixture of desaturators and elongasers all together, together with promoters and so on and so forth. It's a rather heroic molecular biology effort, and put this into either oilseed rape or camelina. These are the main crops which have been used. Soybeans also been modified. This is what happens with camelina. This is the alpha linolenic acid content of wild type camelina, and you modify it with all these extra enzymes here, and it will make long-chain polyunsaturated fatty acids a bit like fish oil does. 15 to 20% is the normal yield, and that's about what you get for the percentage of long-chain polyunsaturated fatty acids in fish oil. So there have been successful modifications of plants, and in the future, no doubt, this will become commercial. Next slide, please. Okay, so just to go back to recommendations on omega-3 polyunsaturated fatty acid intakes, as I said before, it's really the omega-3 polyunsaturated fatty acids that we need to be concerned about. General population, we need about half a gram per day. Individuals with cardiovascular problems, a bit more. And then, of course, there's special requirements for pregnant or lactating females and for infants. Next slide, please. And if I take the ISFAL recommendations, this is the International Society for the Study of Fats and Lipids. They recommend an adequate linolenic acid intake of 2% of energy, a healthy intake of linolenic acid about 0.7% of total energy requirements. That gives you a ratio of omega-6 to omega-3 fatty acids of about 3 to 1, which is reckoned to be a healthy intake. And for cardiovascular health, we need a minimum intake of half a gram per day, and of course a bit more is recommended. So that really takes us up to Philip's presentation. Next slide, please. And thank you for your attention.
Thank you so much, John. So now I'd like to introduce our second speaker, Professor Philip Calder, who is head of the School of Human Development and Health and Professor of Nutritional Immunology in the Faculty of Medicine at Southampton University. He's an internationally recognised researcher on the metabolism and functionality of fatty acids, with an emphasis on the roles of omega-3 fatty acids. His research addresses both life course and translational considerations, and it includes research in cell and animal models, as well as in healthy humans and patients. So over to you, Philip.
Great. Can you see my screen okay?
Yeah, but it's not on full presentation.
Thanks.
That's fine.
Thanks very much.
Fine.
Yeah, excellent. Thanks very much for your kind introduction. And thanks very much, John, for your presentation. My talk is going to build very much on John's and actually sort of go full circle. So I'm mainly going to focus on these long-chain, highly unsaturated omega-3 fatty acids that John has said a little bit about: EPA, eicosapentaenoic acid, DHA, docosahexaenoic acid. These are long-chain, 20 or 22 carbons in the chain, highly unsaturated, 5 or 6 double bonds in the chain. So this long chain and high degree of unsaturation gives these fatty acids unique physical properties, and it also gives them unique functional properties, that is, unique physiological properties. And in fact, the structural and physiological properties are linked to one another, as I'll show you. As John has indicated, EPA and DHA are sort of towards the end of the omega-3 fatty acid biosynthetic chain and can be produced, at least in theory, from the essential omega-3 fatty acid alpha-linolenic acid, which has 18 carbons and 3 double bonds. And as John indicated, this conversion involves desaturation and elongation enzymes. As John mentioned also, there is an analogous biochemical pathway involving the omega-6 fatty acids, with omega-6s on the left here, omega-3s on the right, and the competition at each of these stages of the pathway for the same enzyme between those two families. So they're directly competing with one another for conversion. And what that means is we need probably some sort of balance between those fatty acid families for optimal metabolism of the individual fatty acids, and therefore optimal functionality of cells and tissues. Now, it turns out that EPA and more particularly DHA are not very well synthesized in humans. So in other words, this pathway doesn't really work very well in humans. Now, one reason for that could be the abundance of omega-6 fatty acids in the diet, meaning that this pathway is just losing out in the competition. Whatever the reason, this suggests that preform, that is dietary sources of EPA and DHA, are important if those fatty acids are important to our physiology and to our health. Now, as John has mentioned, EPA and DHA are found in seafood, especially in what we call fatty or oily fish, but also in fish oil supplements, lean fish liver oils like cod liver oil, algal oils, which John touched on, also in the oil of a crustacean called krill, and there are even pharmaceutical preparations of these fatty acids where they're highly concentrated, making up as much as 90% of the preparation. And doctors can actually prescribe these pharma-grade omega-3s for some clinical settings, like the control of hypertriglyceridemia. Let's look at the amounts of these fatty acids in different fish. EPA in light blue, DHA in dark blue. You see the amounts of total EPA plus DHA in fish. This is in 100 grams. That's sort of a small adult portion, I guess. The amounts of these fatty acids varies according to the fish type. In general, fish contain a fair amount of DHA compared to EPA, but in fact, all fish contain some EPA and DHA, but we make this division between the fatty or oily fish, which are highly abundant in these fatty acids, and the white or lean fish, which are relatively low in these fatty acids. But just keep in mind, from eating one meal of fatty fish, we could be getting 1, 2, or maybe even 3 grams of these fatty acids. One meal of lean fish, we might be getting 200 or 300 milligrams. Let's now look at supplements. These are the amounts of EPA and DHA per 1 gram of supplement. A typical supplement you might get from a chemist shop is probably a 1 gram capsule, could be a ½ gram capsule. These are the amounts in a 1 gram capsule. Again, most capsules contain both EPA and DHA, and you see the amounts in standard fish oils, cod liver oil, krill oil are 200 or 300 milligrams in a capsule. That means there is 700 or 800 milligrams of other fatty acids, but then we have fish oil concentrates, 45 or even 60% EPA plus DHA, and then these pharmaceutical preparations, which are 85 or 90% EPA or DHA. But also, again, keep in mind, you need a lot of these capsules to get the amount of omega-3s that you would get simply by eating 1 meal of salmon, 1 meal of sardines, something like that. Typical intakes of the different polyunsaturated fatty acids in Western populations. These are actually average UK data. Linoleic acid is about 10 grams a day. The highly unsaturated omega-6 fatty acid, arachidonic acid, about ½ a gram a day. 1 gram of alpha-linolenic, and in most people, perhaps as much as 0.1 grams of EPA plus DHA per day, but this will be higher in fish eaters and higher in people using fish oil supplements. And of course, it will be lower than this in vegetarians and vegans, and that's been pretty well documented. Now, in both fish and in capsules, in supplements, EPA and DHA are mainly in the triglyceride form. So they're handled like other dietary fats. They're emulsified, they're hydrolyzed in the gut, they're taken up and eventually appear in the bloodstream in lipoproteins. They're transported in lipoproteins as triglycerides and cholesterol esters with phospholipids in the lipoprotein coat. These lipoproteins are delivering fatty acids to different tissues. For example, they might deliver them to adipose tissue for storage. They might deliver them eventually to the liver for metabolism. That could be this chain of metabolism from alpha-linolenic acid to EPA, for example. The liver is an important place for that pathway, even if it's not very active. There's fatty acid oxidation in the liver, of course, but also they deliver EPA and DHA to different cells and tissues where the fatty acids are incorporated into cell membranes. If we look at how much of these fatty acids is in the membranes of different cells. This is the sort of picture we get. These are human immune cells taken from the blood of healthy volunteers, a study led by our chairman or chairwoman today. Now, this is an analysis of fatty acid composition by gas chromatography. All of the fatty acids present would add up to 100%. So immediately you see there's quite a lot of omega-6 fatty acid in the cell membrane, and there's rather less omega-3 fatty acid. So there's around about 30 to 35% of omega-6 fatty acids and maybe 3% of omega-3 fatty acids. And this tenfold difference is actually similar to the tenfold difference that we see in the diet. So the diet is at least in part reflected in the composition of our cell membranes. Now that's immune cells. This is a depiction of data taken from the literature showing the EPA content of different blood lipid fractions, blood cells like red blood cells, platelets, white blood cells, parts of the brain, cerebral cortex, the eye, different muscles and other tissues, including sperm. So you see immediately that in humans, the amount of EPA is quite low, but it's highly variable between different tissues, reflecting perhaps that they have different needs for EPA. If we look at DHA, we see a similar pattern, high variability in different locations in the body, but some locations like parts of the brain represented by cerebral cortex, the eye represented by retina, and sperm having pretty high amounts of DHA compared to other tissues, suggesting that DHA is really important in brain function, in eye function, so visual function, and in sperm function. And indeed, research shows that the DHA content of the brain, the eye, and the sperm is highly related to the function of those specific cell and tissue types. But you see the scale on this graph is rather, goes up about tenfold higher than the scale on the EPA graph. And when we put them together, you see that all places in the body contain much more DHA than EPA. So maybe DHA is of special structural and functional importance compared to EPA. So what happens if people increase their intake of EPA and DHA? So this is data from one of our studies in human volunteers, where we gave people either placebo with no added EPA and DHA, or we gave them one of 3 different doses of a supplement containing EPA plus DHA, and they took these supplements every day for a year, and they came into our clinical facility 9 times during the year, and they gave us blood samples, and we measured the amount of EPA and DHA in different blood pools and also in some other places like adipose tissue. This is the sort of data we found wherever we looked. This is for platelets, the amount of EPA at the top, the amount of DHA at the bottom, and you see both EPA in the top and DHA at the bottom go up over time, and they reach a new plateau that's determined by the dose of the fatty acids that's consumed. You'll see in the placebo group, there's no change over 1 year. So these fatty acids are very stable without any other change. But the important thing I'm showing you is this dose response incorporation of both EPA and DHA, and also that there's a time course. These fatty acids accumulate over a period of weeks to months, depending upon exactly the place you're looking, because that seems to be determined by the turnover of cells in the location. I mentioned dose-response relationship, and this simply shows the dose-response incorporation of EPA plus DHA, so added together, in plasma phospholipids, cholesterol esters, free fatty acids, triglycerides, in white blood cells and in red blood cells in that study, according to placebo as 0, and then these 3 different doses, 1, 2, and 4. And you'll see there's a significant, clear, linear dose-response relationship in each of these places. So in other words, the more you consume, the more that ends up in your blood cells, and indeed in your tissues. Now, so far I have concentrated on blood lipids and blood cells. I've shown you platelets, I've just shown you white cells and red cells, but this is a study looking at the heart. So in this study, they took a small biopsy of hearts that were going to be transplanted into individuals. Then when those individuals received the transplants, they went into a supplementation study where they consumed a modest dose of EPA and DHA every day for 6 months. Then after 6 months, the researchers took another biopsy of the heart. So the blue is EPA or DHA before transplantation, and the red is EPA or DHA after transplantation. So you see that over the 6-month period, both EPA and DHA increase in the heart of these individuals, just like they would have increased in the blood, in the white cells, in the red cells. They increase in skeletal muscle, in the liver, and so on. Now, this increase in the amount of these fatty acids obviously means some other types of fatty acids must be moved out. And in fact, commonly, these are the analogues of the omega-6 family. So in other words, we're getting a switching of omega-3 and omega-6 fatty acids in the membrane. So we could consider this to be a sort of a rebalancing of membrane polyunsaturated fatty acids. Now, this turns out to be really important for how EPA and DHA influence our physiology and impact on our health. So when we increase their intake, they're incorporated into cell membranes. I've shown you data for that on different types of cells and different tissues. Because of this unique physical properties of these fatty acids, particularly their high degree of unsaturation, they influence the structure and the physical nature of the membrane. They influence the environment in which proteins function in the membrane, transport proteins, receptors, enzymes, ion channels in the heart, for example. And they influence the lipid mediators that John mentioned, the prostaglandins, the leukotrienes. There are special mediators made from EPA and DHA called pro-resolution mediators that switch off inflammation and promote our immune response. Let's call these a type of signal generated from membrane phospholipids. So with these differences in structural aspects of the membrane protein function, and the lipid mediators being produced, membranes enriched in EPA and DHA send different signals than membranes that are poor in those fatty acids. Those signals influence gene expression, protein production, and cell and tissue responses. And this changes, if you like, the physiology of cells, the physiology of tissues, in a way that seems to be related to improved health and improved recovery from some diseases. So through these mechanisms, EPA and DHA influence visual function and cognitive function. DHA is really important in early development of the brain and of the eye, and that's why the recommendations for pregnant women and lactating women are higher than the recommendations for other adults in the population. These fatty acids influence our metabolism. I've mentioned that they can influence inflammation and immunity, oxidative stress, blood coagulation, and the function of many different tissues, wound healing, and lots of other things. So what do they actually do? I've mentioned DHA, brain growth, visual and neural function, cognitive mental function. I've mentioned their importance in membrane structure, and they also do things like help control blood pressure, blood clotting, blood triglycerides, blood vessel function, heart rhythm, and other things that I've mentioned, bone health, insulin sensitivity. And that means they can help with lots of things that occur in the population. So these fatty acids, EPA and DHA, are really important for optimal function and health right across the life course. I've mentioned in passing pregnancy, lactation, and infancy, but of course, many of the other things I have on the slide here occur as we get older. So these fatty acids are really important right through middle age and into old age, so right across the life course. As a result of their effects, there are actually approved health claims for EPA and DHA in Europe. So the approved health claims are that they contribute to maintenance of normal blood pressure, normal blood triglyceride levels, and normal function of the heart. So the evidence around these functions is really, really strong, and that gives manufacturers the ability to make these substantiated health claims that are covered by legal regulations. How much do we need? John mentioned that there are different recommendations in different countries, but they're reasonably similar to one another. 250 milligrams is adequate unless you're pregnant, in which case you need more DHA. That's the European recommendation. FAO is similar to that, at least 250 milligrams per day, but more than that if you're pregnant or lactating. In the UK, it's at least 450 milligrams per day, preferably from fish. Personally, I feel these recommendations are a bit conservative, and I would say we probably need at least 1,000 milligrams per day. So in summary, the roles and mechanisms of action of EPA and DHA have been widely studied. Intakes of these fatty acids are usually low, but they can be increased through the diet, particularly oily fish, or the use of supplements. And it's important to note that most people actually don't meet the recommendations for intake that was shown on the previous slide. I've shown you that increased intake of these fatty acids is reflected in higher levels in the blood and in tissues, and that this results in modification of cell and tissue function in a way that's consistent with improved health across the life course. So, you know, the bottom line is eat fish. Or if not, to get these fatty acids, you need to think about supplements. But I would prefer that people ate fish. The problem, of course, is that fish and fish oil supplements are not sustainable. So we really have to explore other sources. John has mentioned algae and algal oils for EPA and DHA, and he touched on the possibility of genetically modified plants being able to produce EPA and DHA, which would be a sustainable terrestrial source of these fatty acids. Another approach is actually to think about the biochemical pathway and try to find out why the pathway doesn't work so well in a lot of humans, and to develop strategies to make that pathway more active. Those strategies might include lowering linoleic acid, the use of micronutrients, because these are cofactors for the desaturase and elongase enzymes. These are the sorts of things that we're working on in my group at the moment. So watch this space for interesting development in these areas. Thanks very much for your attention.
Thanks very much, Philip. And I think shortly we're going to have John back for a brief period of questions. So there have been some questions in already. The first one I think John actually answered, which was about whether terrestrial plants produce essential fatty acids or is it just algae? And I think John explained that in order for terrestrial plants to produce long chain omega-3s, you need to genetically modify them. That's correct, isn't it?
That's right, for higher plants, yeah. Algae, I mean, there are some lower plants, as they're called, but none of the crop plants produce longer chain fatty acids. So that's where algae come into their own. Actually, algae are already used commercially to produce, for example, infant formula milks. Schizochytrium is already used that commercially.
And also on the biochemistry, just to confirm that mammals don't have the ability to convert omega-3s to omega-6 or vice versa, and that's why you have competition between those two pathways, isn't it?
Yes, indeed. Yeah, they lack the two desaturase enzymes, first of all, to generate the omega-6 series of fatty acids, and then to convert those to the omega-3. So we lack those two desaturases.
And John, there's a question for you from Hadika, who says, could you clarify if the delta 8 pathway and the delta 6 pathway are both present in algae, and what did you mean by conventional delta 6?
Well, conventional delta 6 or omega-6 fatty acids, I'm referring to linoleic acid, which is present in our diet in quite large amounts. As Philip mentioned, you know, we usually, or most Western diets, contain too much omega-6 fatty acids in our diet. In fact, it's ironic that fluoromargarine, for example, which is made from sunflower oil, was touted as being very good for your health. And then suddenly they realized that taking in too much omega-6 fatty acids, which are present in sunflower oil, which is then converted to fluoromargarine, people were taking in too much, and that was realized later on. And so if you look at the new fluoromargarine packets, they don't emphasize the sunflower oil content quite as much.
Thanks, John. There's a question here from somebody you might know. It's Keith Frayne, who's textbook on metabolic regulation should be on every biochemist bookshelf. So Keith asks, if freshwater algae don't make long chain PUFA, that is the 20 and 22 carbon fatty acids, then do wild freshwater trout have them?
Yeah, well, I did mention that there are huge numbers of algae, and this means, of course, that you can always find one or two freshwater algae that do make these very long chain fatty acids. Most of them don't, but there are exceptions to that. So those were generalizations that I mentioned. And in fact, if you look at wild trout which are farmed which are caught in the UK, they do contain small amounts of omega-3 polyunsaturated fatty acids. It just depends which algae they eat, and Philip showed some data for individual fish, and depending what fish eat, of course, that then actually determines what the ratio of EPA to DHA, etc., that they will accumulate.
Philip, do you want to add to that?
Yeah, I think a very important continuation of this point is that fish are just like us. They're also not very good at carrying out this metabolic pathway, and they're reliant on their diet, just like we're reliant on our diet. So I think this is really passing the algal fatty acids through the food chain. And I think, John's emphasis on algae is therefore quite important in terms of the context of, where we're all going to get these fatty acids from eventually.
Thank you. And a question for you, Philip. So is there, sorry about the drilling if you can hear that, is there data to directly support the association between DHA and EPA content of membranes and the positive impact on signaling pathways and physiology? So between the biochemistry and the physiology?
Yeah, absolutely. So, the research on these fatty acids has been being carried out, I think seriously since the early 1980s. And there's a lot of work in model systems and also in humans that link effects on signaling pathways. I'm most familiar, of course, with things around inflammation. So, for example, the NF-kappa B signaling pathway, which is influenced in a concentration-dependent manner by DHA. Actually, it turns out it's DHA in the membrane that is modulating this pathway, and that relates to the anti-inflammatory effects of omega-3s that you might see, for example, in a person with an inflammatory disease who's treated with omega-3s to try to control their inflammation. So I think there is a direct link which you could put together by looking at the literature between incorporation into the membrane, the signaling pathways, what the cells are doing, and what their benefit or the effect on health is, for sure.
Thanks. And a question from Hannah Dallas, who says, have you looked at the potential of single cell microorganisms as potential sources of fatty acids? And by that she means not just algae, but bacteria or fungi.
That's for John.
John, that might be for you.
Okay, well, we haven't done any research on that, but of course, there has been considerable work, particularly with yeast, single cell yeast, which have been developed to an extent commercially to produce a number of these polyunsaturated 20 and 22, well, 20 carbon polyunsaturated fatty acids. So not myself, but other people have done that. Okay. Colin Ratledge in Hull was particularly prominent with this research.
Yeah. I think, John, one of the reasons people have focused on algae is what you indicated, is you can make algae accumulate lipid and then release it. So you can sort of grow algae in, this is what they do for the infant formula industry that John talked about. You can grow algae in vats, and they're just churning out this lipid that you sort of skim off and then clean up and use. And it may be that some other organisms don't quite do it like that, although I'm not so familiar with those systems.
Yeah.
There's another biochemical question here, which is about why the kingdom Animalia lacks the ability for synthesis of unsaturated fatty acids from acetyl-CoA. It's not that it lacks the ability entirely, is it? It's just that we can't synthesize certain unsaturated fatty acids. Is that right?
So I think,
as I said, we lack the delta 12 and the delta 15 desaturases are not present in mammals or in animals in general. Although 1 or 2 insects can make linoleic acid, but mammals certainly just lack these desaturases.
Yeah.
Now you could ask why do we lack the desaturases? Well, I'm going to answer to that, but we just lack them.
Yeah, that's right. I'm going to ask one final quick question. Well, it's not really a quick question, but let's see if you can give me a quick answer. So Rania thanks you for great talks and asks what you think of olive oil and walnuts as sources of omega-3 and omega-6.
Well, olive oil contains very, very low levels of omega-3 fatty acids. In fact, for a high oleate oil, oilseed rape oil is very good. It's got a very good ratio of omega-3 to omega-6 fatty acids. Of course, it doesn't taste nice. It's not like olive oil, because olive oil contains phenolics, which give it its flavor. So olive oil is no good as a source of omega-3 fatty acids. It's got a reasonable amount of omega-6, but if you want omega-6, there's no problem getting that. As I said, sunflower oil has got 70% or so of linoleic acid. Walnuts, yeah, they're a source of omega-3 fatty acids, and good in the diet. The so-called Mediterranean diet, which has been put forward as being a very healthy diet, of course, contains lots of things, not just olive oil. It contains, lots of fish, lots of fruit, lots of nuts, and so on. And walnuts could be a component there.
Thanks, John. There are still more questions that we haven't answered, but I am going to have to unfortunately draw it to a close. But I would like to thank both speakers for their really clear and insightful presentations, and also to you, the audience, for asking some great questions. You can actually continue the conversation online if you follow at BiochemSoc and at PP Publishing on Twitter. And if you enjoyed today's session and would like to know more about the topic, you can visit the Essays in Biochemistry website for some related reading. As I said, if you have an idea for a webinar, we'd encourage you to submit a proposal. And if you've missed any of the 40+ webinars that we've run as part of the series, or you'd like to watch them again, then you can visit the website or the YouTube channel. So the next webinar in our dedicated ECR series is Developments in Glycobiology on Wednesday, the 19th of January at 2 p.m. UK time. And at that, you'll hear from 3 ECR members of the Society who will share their work in the field of glycobiology. And you might also be interested in the From Golgi Organisation to Glycan Function conference in Brighton, UK, from the 5th to the 7th of April this year. So you can visit the Society's website for more details on that. And finally, it's been great to have you here with us today. It's more important than ever at the moment to stay connected within the biosciences community. Joining the Biochemical Society is a really great way to engage and to take advantage of discounted registration fees for Society courses and meetings, for exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and much more. So please do visit the website to find out more. Thanks again, stay safe, and goodbye.